Good afternoon, everyone, and thank you for attending my symposium talk. This year, I tested the prevalence of anemia and beta thalassemia in Asian adolescent girls. My inspiration in this project began from my initial interest in sickle cell disease, where I found that beta thalassemia is a similar disease which is understudied in medicine. Beta thalassemia's heredity affects a large number of Asians, which has also piqued my interest because my parents are Chinese immigrants. Anemia is an acquired condition which is caused by a lack of healthy red blood cells in the body. Individuals with anemia often lack enough iron in their diet, which results in iron deficiency and an inadequate amount of hemoglobin in their bodies. Hemoglobin is a protein produced by the body that binds with oxygen in order to create healthy red blood cells to oxygenate the body. Anemia, however, is particularly susceptible in women due to a constant loss of iron during menstruation, as well as women not eating enough iron-rich foods like meat or poultry to sustain healthy levels of iron in their bodies. This condition is a perpetuating issue in poverty-stricken countries, affecting over 800 million women within the reproductive age. In extreme cases of anemia, cognitive function, scholastic performance, and physical tolerance are decreased due to this lack of hemoglobin. Beta thalassemia minor is a blood disorder that reduces the production of hemoglobin. Due to the lack of hemoglobin production, the red blood cells aren't able to oxygenate the body. In result, anemia may also be a symptom of thalassemia due to a lack of healthy red blood cells in the bloodstream. Beta thalassemia is a hereditary blood disorder, which means that it is passed down from a carrier of the gene to their offspring. Previous studies show that beta thalassemia is highly prevalent in many Southeast Asian countries such as Indonesia, China, and Vietnam. Beta thalassemia comes in 3 degrees of severity: minor, intermediate, and major. The degrees of severity are diagnosed based on the complexity of the mutations to chromosome 11, inherited by the child of the carrier. Beta thalassemia minor exhibits few or no symptoms of fatigue, bloating, and anemia. Beta thalassemia intermediate symptoms include moderate to severe anemia, extreme fatigue, delayed growth, weakened bones, or an enlarged spleen. Beta thalassemia major is the most severe degree of thalassemia and results in a change in shape of the bones in the face and the head. Children born with thalassemia major typically do not live past the age of 17 and die from heart complications. Because of beta thalassemia minor's shallow symptoms, it is often mistaken for iron deficiency anemia. This serves a big issue due to varying treatment plans of an acquired illness and a hereditary disease. Iron pills are the most common treatment for anemia. However, iron pills cause iron overload to individuals with thalassemia due to the pre-existing lack of hemoglobin to process this iron supplement. In result, the medication is detrimental to the body and does more harm than good. Thalassemia heredity poses a threat to Hawaii's community by the undercoverage of prevalence within our community. In addition, Hawaii's Asian population continues to grow with little pre-screening protocol. As you see on the screen, carrier parents that have beta thalassemia minor may be asymptomatic. However, they have a 25% chance of a child inheriting thalassemia major, a 50% chance of a carrier child with thalassemia minor or intermediate, and a 25% chance of a healthy child with no affected genes. There is no instant cure to beta thalassemia besides bone marrow and stem cell transplants, which require a specific donor and are extremely expensive. A study in 1991 by Jana et al. Studied the rate of beta thalassemia carriers in Hawaii, finding a starking 34% of Chinese, 55% of Laotian, and 40% of Filipino people held carrier genes to this disorder. This led me to my research question of how prevalent is anemia and beta thalassemia minor in a group of Asian adolescent girls. The goal of this project was to be able to quantify the prevalence of anemia and the prevalence of beta thalassemia through a more susceptible test group, Southeast Asian girls. The first thing I decided to test for was anemia to find the prevalence of low hemoglobin levels along my participants. I used a hemoglobin monitor to read their hemoglobin levels by drawing a drop of blood from their finger using a sterile lancet. Similar to a glucose monitor, this machine is able to read 2 crucial measurements: the hemoglobin level and the hematocrit level, which is the percent of healthy red blood cells within the sample. By doing so, I was able to set a benchmark of hemoglobin levels for my Southeast Asian cohort of girls. Results from this test revealed surprisingly low hemoglobin levels. Out of the 11 participants, 7 out of the 11 girls were below the lower bound of a healthy individual, which is above 12 grams per deciliter. 10 hematocrit samples were also below the lower bound of 36%. These results supported a UH study done in 2012 of anemia prevalence in Hawaii, revealing 45% of participants testing anemic. In addition, 31 females and 14 males tested anemic, which again validates the statement that females are more prone to anemia than males. The second part of my project was to detect the prevalence of thalassemia in adolescent girls. The conventional way to test someone for beta thalassemia is through a method called hemoglobin electrophoresis. This method was more mainstream and produced a quick result of a panel of sickle cell diagnoses. However, equipment for this procedure was extremely expensive, which made this traditional method infeasible in a high school lab. This presented a new research goal to develop a PCR sequencing method that can detect beta thalassemia mutations. I was faced with a panel of PCR methods such as the ARMS and multiplex PCR procedures, and spent nearly 5 months of trial and error before I finally was able to successfully identify a thalassemia carrier in February. I decided to try another PCR method and sequence. DNA was extracted from a blood sample using the spin column method. The template of DNA was combined into a mixture where specific primers designated the parameters of the gene, metaphorically acting like fences to designate where a lawn can be mowed. The samples combined with the primers were placed into a thermocycler machine where temperatures fluctuated to amplify any mutations between the designated regions set by the primers. Once the samples have been amplified for any mutations, they are sent off for sequencing. The image on the screen is a display of the results from the sequencing. A reference gene was pulled from GenBank, which represents a healthy human comparison to the samples below. The specific base pairs of the HBB gene, which is a protein coding gene, was able to be compared to the exact same location of the other 2 participants. By doing so, mutations can be located by finding the discrepancies between the reference gene, the non-thalassemia gene, and the thalassemia gene. In result of the gene mapping, mutation IVS2654 was located on a carrier sample of a participant. We were able to identify the exact location on the HBB gene through the gene mapping process. Broken down into diagrams, the HBB gene has 3 exons. Take note of the highlighted 18 and 19 primers. These primers were used to designate the range of detectable mutations when sequencing through PCR. The image below is able to give us more context of where mutation IVS2654 sits. As we zoom in between the primers 18 and 19, we see the IVS1 and IVS2 regions. Zoom into the IVS2 region and we find IVS2654 at the 654th base pair of the specific region. However, in the grand scheme of the entire HBB gene, IVS2654 is located in the 1149th base pair of the gene itself. Now, the occurrence of this common mutation happens by a change to chromosome 11. As you see on the image to the left, the box mutation shows a mutated base of a C to a T. In addition, the image to the right is a zoomed-in version of the gene map. The double-colored hump on the YCB-BT sequence reveals one mutation to the chromosome, meaning one was mutated to a C, showing heterozygous chromosomes. Chromosomes come in pairs, therefore, because this participant was only a carrier of the gene, one mutation, which is IVS2654, is displayed by a double hump of blue and red. In a more severe case, someone with 2 gene mutations may have an entirely different single-colored hump due to 2 mutations being present. In a base pair sample of around 900, we were looking for this small change in expression, which revealed that my PCR sequencing method was a success. In hindsight, a lot of troubleshooting was done to be able to test for beta thalassemia minor, including a genetic PCR method. A mass of the 5 months was spent optimizing the concentrations of the primers. An Anchor article of newborn screening for beta thalassemia was used to create our primers for adolescent screening. A primary issue we faced was primers setting a parameter too small to detect a mutation, when I then realized that by setting a larger parameter, more mutations can be detected. Once primers were switched, I was faced with nonspecific bands, which are the amplification of the wrong part of the DNA, creating a false positive. Nonspecific bands occur in the thermocycler where temperatures are inadequate for amplification. To troubleshoot, I ran multiple gradient tests of a range of temperatures of the same DNA to optimize the environment for DNA to be successfully amplified. In summary, I found a high prevalence of anemia among adolescent girls and was able to detect a beta thalassemia mutation through sequencing. I was able to detect one of the most common Asian mutations, IBS2654, through a method other than hemoglobin electrophoresis, therefore fulfilling my research goal of developing a new way around the conventional pre-screening method. I found that the prevalence of anemia among a Southeast Asian cohort of girls was surprisingly prevalent with low hemoglobin levels across the board. This gives evidence that Southeast Asians are particularly prone to acquiring anemia in the span of their lifetimes, already testing low while maintaining healthy lifestyles and diets. My project has much potential to provide valuable information about hereditary prevalence in Hawaii's community. Future research includes testing all of my participants and extending the criteria to males and different ethnicities. By doing a holistic study of beta thalassemia on gender and ethnicity in Hawaii, we can compare the perpetuation of beta thalassemia with Southeast Asian countries. Locally, I would also push for more pre-screening protocols. The beta thalassemia minor carrier in my project did not let it stop her from running a marathon. However, she was only aware of her mutation when she took a blood test after she became pregnant. I would like to see more precaution for this hereditary disorder in our community, given that we are the state with the highest Asian population in America. Throughout my 2 years of research, I have learned many things about not giving up and trying again. Though it can be frustrating at times, the reward of presenting your findings is immeasurable to all the suffering you endured. My advice to future researchers is to choose something you're passionate about and to know that the data you discover is helping your community. And if any of you here are considering research next year, I think that this is the best class Iolani has to offer. Lastly, I'd like to give a big thanks to friends, family, and Iolani for the constant support. Thank you, Dr. Chan, for being an amazing mentor and teacher, and for Ms. Kobayashi and Mr. Hill for helping me every step of the way and calculating every failed master mix. Thank you.